Good afternoon. I'm Dr. Williams. For those of you who are online and can't see us yet, but it's my pleasure to introduce our next version of Research Grand Rounds. It really is a delight to have someone who is in our department, but not completely of it. It's Dr. Sham Kakar. He is a entrepreneur, as I'm learning more about him, and a Ph.D. He actually hails from India originally, but he first joined the faculty as an assistant professor in India, and then he moved to the United States in Toledo, Ohio, as a research assistant. Worked his way up through University of Alabama, Birmingham, in their comprehensive cancer center, and he came to University of Louisville in 2000. So this would be your 24th year. Congratulations! Next year, 25. And he actually is in both the Department of Medicine and the Department of Physiology. And if you look through his CV, you see hundreds of publications, millions of dollars of grant funding, so much leadership, and so much entrepreneurship. And so we're very proud to have him as an MBA from our esteemed institution in the College of Business on entrepreneurship. And he'll be speaking today about cancer-induced cardiac cachexia, and it's reversible. Hopefully it is very reversible. Thank you so much for doing this.
Thank you. Thank you, Dr. William, for your nice introduction. So this is something new I learned a few years back. Never heard about cachexia. So I hired a postdoc, Dr. Alex Tragan, who did Ph.D. from University of Louisville from Department of Anatomy in Muscle Biology. So he came to my lab, and this was funded by Maurice and me under T32 training grant. He said the drug you are using, which I'll describe, let's test on the muscle weakening, which is called cachexia. So I accepted his concept, and we start doing it, and we build up a lot of publication and a lot of work on this aspect. So my initial training has been in reproductive endocrinology, and I came across, I worked with several endocrinologists, well-known, world-renowned scientists in this area. So if we look the top one, some of this pointer can see the red here, but I cannot do there. So Andrew Shelley, he was the first one to discover GnRH or LHRH peptide. This is stand for gonadotropin releasing hormone and luteinizing releasing hormone in early 70s. So he shared Nobel Prize with his mentor, Roger Gilman, in 1977 in physiology and medicine. Then next was Dr. Ernie Nobel. He was chairman of Department of Physiology at Pittsburgh, and he followed Dr. Andrew Shelley and interacted with him. What he discovered that GnR excretion from the hypothalamic neuron is pulsatile, which is a basis for the treatment of precocious puberty and androgen-dependent prostate cancer. The third on the ladder was Jimmy Neal. I worked directly with him as a junior faculty. He was chairman of Department of Physiology at UofL. What he found out that what types of cell in the pituitary secrete GnRH or LH hormone, and he was first one to find out how estrogen and progesterone level are regulated in estrocycle in mouse and rats, and in menestrocycle in human. Then I come at the fourth place on the ladder, but I learned a lot of them from them, and we know 80s and 90s were the era of molecular biology. So everybody was trying to clone some gene. So I end up cloning several genes. The first one I clone is cretogranine 2, which is a secretory protein, which helps in packaging of secretory proteins such as the hormones and other secretory proteins. And my early work, I focus on cardiovascular research at Toledo, and I end up cloning angiotensin 2 receptor which is a major regulator for blood pressure regulation, which was a big breakthrough in the field of endocrinology. Then I end up cloning GnRH receptor, which is down the ladder. And then I clone PTTG, which stands for pituitary tumor transforming gene, and also known as scurin. This is an oncogene, and I put a lot of efforts on this cloning and was really fortunate to get several grants and establish myself as a independent investigator. So after I cloned GNR receptor, I shifted to cancer research. And I said recently, I learned about what cancer can cause to the other organ in the body. If you look this figure, cancer not only damage the tissue where it's originate, but several tissue in the body, which include adipose tissue, liver, gut, skeletal muscle, brain, heart, blood vessel, pancreas, bone, and testes, basically all the tissue and the organ. The major tissue which are affected, and most of the people have studied, is the skeletal muscle as well as cardiac muscle. So both these organs are very much affected, and that defect or dysfunction of this organ is called cachexia. That's why I title is cancer-induced cachexia. So what is cachexia? It's a loss of body weight, voluntary loss of body weight, 5% after the cancer is initiated. It happens 42 to 80% of the patient with cancer and directly responsible for 30% of the death. Cancer cachexia is associated with weakness or loss of muscle, fatigue, and significantly impaired quality of life. You must have seen patients with cancer, they cannot walk, they feel very bad, they cannot eat, they lost the appetite. That's because of induction of cachexia. And the other symptoms are there, but the good part is that, or bad part, I can say, so far, there is no FDA approval drug for this disorder or dysfunction. So in that respect, I target my research to find out if we can develop some drug which can target cachexia, because this is a big problem in cancer patients. So start working on vidofludimus A, which is I've been working for a couple of years, and vidofludimus A is a purified product from the plant called ashwagandha. And it has an anti-cancer property as well as anti-inflammatory property. We, as well as many other investigators, have defined its major role as an anti-cancer drug. So we set up the cachexia model in mouse, NSG mice, and this is the plant. So we have a control versus the tumor bearing. The way we use this tumor model, which is based on my lot of publication, we inject the tumor cell, ovarian cancer cell, into the peritoneal cavity, which develop metastatic tumor, which is a major cause for death of the patient with ovarian cancer. So keeping in my own field, this is some introduction about vidofludimus. As I mentioned, that this is purified from Vidania somniferea. Another name is ashwagandha or winter cherry, and this is the chemical structure, and it's well established as anti-cancer drug. So this was the experimental plan. So we have animal, and after 1 week of getting the animal, we did the grip strength, which really tells how strong the animal are. And then after 1 week of getting the animal, we injected the animal with the ovarian cancer stem cell. And after a week of injection of tumor cell, we chased them with the vidofludimus A. And this was done by intraperitoneal injection. So initially we used 2 doses, 2 milligram per kg and 4 milligram per kg. The injection of WFA was administered every 3rd day and carried out 3 weeks after the 1 week of injection of tumor cell. So this is a grip centimeter, and we can see it has a bar and it has a grill. So to look the 4-limb grip stand, we allow the animal to hold the bar and then pull from the tail. And the force required for the animal to pull, release the bar is called grip strength, four-limb grip strength. In case of all four limbs, our total grip strength, we have a grill, let the animal hold the grill with all the four paws and then try to pull it. Strength required is much more here compared to four limb. So in all the animal we use in our experiment, we did not see much difference from one animal to another. So after 1 week of injection, just 1 week of injection of tumor cell, you can see the big difference in forelimb as well as total grip strength. It was highly significantly reduced just in 1 week. And when we chase the animal with vidofludimus after 1 week of injection of tumor cell, you can see that in control animal, we see improvement in grip strength. And this is because vidofludimus A is also known to build up the muscle. And in our experiment, we showed that there was an increase in differentiating satellite cell. These satellite cells are the stem cell in muscle, which differentiate into muscle cell and increase. We saw the increase in the satellite cell, which could be the reason of building the muscle. And with the animal which were bearing tumor, again, it was significantly reduced just after 1 week or 2 weeks from the injection of cells. And then it was almost attenuated in both cases on treatment with either dose of vidofludimus A. So which was exciting, and the first experiment we did, and my postdoc was very happy that his theory was really workable. So we looked for 4 weeks just before sacrificing that. We did a third week, but I'm not showing here. So same thing, we saw a significant drop in grip strength, forelimb as well as total grip strength. And again, it was attenuated in both cases by either dose of vidofludimus A. And the attenuation was highly significant compared to the tumor-bearing animal that were treated with vehicle only. So we look at tumor growth. You can see there is a body weight loss, which is a sign for developing cachexia, and it was attenuated with WFA treatment. Tumor size was increased, which is shown here also. Vidofludimus reduced the tumor size. And when we inject the tumor into peritoneal cavity, it metastasized to the ovaries. The ovary size and the weight was increased, and treatment with WFA did not completely brought to the normal level, but significantly suppressed the tumor growth and the weight. So to confirm that it was happening at cellular level, so we did the H&E staining of the muscle tissue, and we can see in control, we have this muscle cell. And then when we treat with WFA, we can see the cell size increase, that means cross-sectional area of the cell increase. And if we look the tumor-bearing animal, the cell cross-sectional area was significantly increased, and then it was brought back to the normal level on treatment of WFA. Here is a quantitation. So as I said, WFA by itself increased the muscle weight, which is due to increase in the cross-sectional area of the muscle cell, and tumor-bearing animal cross-sectional area was significantly reduced, which was again attenuated with WFA treatment. Minimum Farad diameter is the length of the muscle fiber. So we did measurement of those one, and they replicated what we saw in the cross-sectional area. So we did we studied a number of mechanisms that are known to induce cachexia, but I'm not showing here because there are several, and if I go just on this one mechanism, I can finish my lecture, but I want to talk to you more about the cardiac cachexia. So we know tumors secrete a lot of growth factor, hormones, cytokines, and PAM and DAM. PAM stand for pathogen associated molecular pattern in cancer. damage associated molecule. So when they bind to the receptor, also the ATP, they activate the formation of inflammasome. And the most common inflammasome are NLRP3, caspase-1, and ASE. And when they get together, they form inflammasome and activate caspase-1. So caspase-1 inactive is converted into active caspase-1, and caspase-1 converts the pro-interleukin 1 beta to active IL-1 beta, and same thing pro-IL-18 to active IL-18, which goes to the membrane and then affect the other cells in the body. So this is just a summary of the inflammasome, but we study a lot of other mechanisms, that protein degradation mechanism, UPS, and ubiquitin pathway, as well as autophagy pathway. So putting things together, this is what we did in our first manuscript, that there are several factors coming from the tumor. They induce stress, cell interest, change the calcium homeostasis, change the protein synthesis, and activate TRAP6 pathways, and autophagy pathways, UPS pathways, and vidofrin. What we observed was inhibiting all the activated pathway by tumor. So this explains how vidofrin works by targeting different mechanisms which induce skeletal muscle atrophy. Now, during that time, I had another postdoc under T32 training grant, Nadia Clem. She got Ph.D. in physiology and had experience on cardiovascular research. When she joined the lab, and she said, well, cardiac is muscle too. So let's look the cardiac muscle and cardiac function. So we decided to carry on the cardiac. So what we saw, that if you look here, this is the control animal, the heart size, and which was a little bit increased or maintained by vidofrin treatment. And in tumor animal, the heart size decreased. The atrophy of the heart was there, and treatment with WFA bring back to the almost normal weight of the heart or the size of the heart. So we did the cardiac function by echocardiography to look for the diameter, fraction shortening, and so on. So if you look here, that's beating normally. This is just a echocardiograph of the animal heart. And then in this case, you can see, so based on this data, we analyze the fraction shortening, ejection fraction, diameter of the heart, and so on. So if we look the heart here, as I said, the heart size was decreased. So the weight of the heart was decreased, which was normalized to the tibial length. Tibial is a long bone and it doesn't get affected by drug. So it was normalized to tibial length. Left ventricle diameter was decreased because there was an atrophy of the heart, which was normalized with WFA, and cross-sectional area also decreased, which was again normalized by WFA. So this is another, if you look here at the heart rate, beat per minute, it was significantly decreased in tumor-bearing animal and was attenuated WFA. And same thing, fraction shortening was decreased significantly, less than 40. Normally it's a 50 or 50 and 60, and it was attenuated by WFA treatment. Cardiac output, same way, was decreased, attenuated by WFA, and LV mass, left ventricle mass, was also decreased since there was atrophy of the muscle, and it was attenuated. So the systolic function was completely restored on WFA treatment, whereas diastolic function of the heart was there, damage was there, but it was not fully restored. We don't know the reason, but one of the reasons could be WFA treatment was only for 3 weeks. Probably it required longer treatment to recover the diastolic function. But it was amazing data and we were very excited. And here, one of the things we found that animal develop arrhythmia. So I'll show you here if this is the control animal. So you can see the normal beating of the heart. We have the regular pulse. And now we got to the tumor bearing animal. And you will see the missing of the beat and stopping of the heart here. And there's a missing of the beat. So if you want to look again, so you can completely see that it was stopped or missed few beats, which could be a reason why patient die from arrhythmia while sleeping. And when we treated this animal with the WFA, either dose, going so fast. So it was restored. There was no missing or no arrhythmia sign in this animal when treated with either dose of WFA. So if you look here, this is the second dose. So we didn't see the sign of arrhythmia. So which tells that the people who are cancer patient or other people who have arrhythmia sign, they can start taking this one as a supplement, and maybe it will be helpful to avoid arrhythmia or cure arrhythmia. So we also looked at diastolic function, A and E ratio, and see this is a very normal, and this is normal here. Now, if you look the calculation in normal condition, A peak is much higher than E peak is much higher than A peak, and treatment with WFA doesn't make much difference. Whereas the tumor-bearing animal, E peak was shortened and A peak was increased, which shows the sign of damage to the heart, which is diastolic function. And if you see the quantitation here, we lost A over A ratio was significantly decreased, but it was never restored to the normal level. And same thing is E over E prime ratio. It was decreased in the heart in tumor bearing animal, but it never came back to the normal. So this is the cross-sectional area of the myocyte. So if you look here, as I said, the normalized heart rate was decreased significantly. Cardiac myocyte cross-sectional area was decreased significantly and attenuated to almost to the normal level. Troponin I, which is a protein which increased the contractility of the muscle, was also decreased. This is the expression level of troponin 1 in myocyte, and then it was normalized by WFA. MHC alpha and MHC beta, many of you are working cardiovascular system. MHC stands for heavy chain protein. So which is alpha is a fetal form, and beta is alpha is a adult form, beta is a fetal form. So the adult form MHC decreased, whereas the MHC fetal form was decreased, and treatment with WFA brought back almost to the normal level. We also look fibrosis of the heart tissue, because fibrosis is one of the major cause for dysfunction of the heart. So we stain the tissue with the Mason trichome. We can see that there was no sign of fibrosis in control animal, and control animal treated with the WFA. Whereas the tumor-bearing animals showed big sign, a great sign of fibrosis, and we measured the area of fibrosis, which is shown here. It was significantly increased and decreased by WFA treatment. And these are some of the things we look. Angiotensin 2 in the circulation was increased. AT1AR, which is a major angiotensin 2 regulator for the heart function, was upregulated, which is known. AT2R, which is a vasoconstrictor, also was increased. TN alpha, AL6, MIP2, which is analog of IL8 in human, interferon gamma, all the cytokines were upregulated in the muscle expression, as well as we measured in the blood their level was also increased, and their level was attenuated by treatment with WFA. These are other ATP which works through P2X7, P2X4, NLRP and inflammasome member was also increased, and they were again decreased by WFA treatment. So this is the mechanism we have put together. It's complicated, so may not go in detail. As I said, tumors create a lot of growth factor, cytokine, chemokines, PAM and DAM. In this case, we are focusing on angiotensin 2, which binds to its receptor and interact with MD2 and activate TRAP6, and then secrete the inflammatory cytokine such as TN-alpha, which activates MAP kinase, as well as NF-kappa B pathways, MAP box as MUT1. These are the major twist. They are the major regulator for cachexia or muscle weakening. So we saw upregulation of those one. And this is the TAM and PAM, how they activate the formation of inflammasome and secretion of IL-1 beta and IL-18. So all these pathways were downregulated when treated with WFA. Angiotensin level went down, cytokine level went down, inflammasome level went down. So this could be a major mechanism by inducing cachexia in heart. So generation of intraperitoneal ovarian tumor in mice results in development of muscle and cardiac cachexia. Treatment of animal with WFA restored the muscle cachexia, including muscle cell cross-sectional area. WFA reduced the cytokine inflammasome generated by tumor as well as muscle and cardiac tissue. Systolic function of left ventricle cachexia was attenuated by WFA. Systolic function was completely preserved in cancer-breeding mice. Diastolic function was improved, but not completely reversed. Arrhythmia was observed in all the tumor-bearing vesicle animal, but not presented in any other group. So very interesting and convincing story, right? So we submitted the grant to NIH R1. The reviewer came back that the effect what you're showing reducing the inflammasome, inflammatory cytokine and repair of heart and all these things could be because WFA is an anti-cancer drug. So what effect you are seeing? Suppression of the tumor, which results in suppression of cytokine, chemokine, and all those factors. Now, how to get around it? Okay, so that was a question. I went to sleep for 30 minutes, which I do normally, almost every day when I have something to think about it. So we came up with another model. As I said, that there was an increase in angiotensin 2 in the circulation and increase in angiotensin 2 in circulation in patient as well as in animal model cause cardiac damage or cardiac dysfunction, which is known. So we generated angiotensin 2 increasing the circulation level. So we took C57BL6 mice, which are immune competent mice, and we implanted the angiotensin 2 pump. In control, we implanted the saline pump. And these pumps are osmotic pumps, so they release angiotensin 2 in a constant manner in a specific dose. So it keeps the level high in the circulation for 4 weeks. So that's the model, is well-established model. So you implant the pump under the skin and let it go. So this is the scheme we did. We implanted saline pump as well as angiotensin 2 pump in the groups of animals. And under saline, we treat with WFA, and also the angiotensin 2 implants, we also treated with WFA. So what we got here, almost same what we saw in tumor. So forelimb, total limb was affected by angiotensin 2 infusion, which was significantly reduced forelimb as well as total, and it was attenuated on treatment with WFA. So that's what This is established, this is well known, and we could repeat the other investigation. Tibial, gastrocnemial, quadriceps, their mass was decreased, and you can see the mass significant decrease, and then it was attenuated with WFA. So this confirmed that model is working, and WFA is effective in recovering the muscle strength and the mass. So if you look at the cardiac heart, so there was a hypertrophy. This is different than what we see in cancer. Cancer, we see the trophy, the heart size goes down. But in this case, the heart size goes up, but the dysfunction induced by angiotensin II mimic the cancer dysfunction. So this is a very good model to compare. So the heart, the size was increased, and when we normalize with the tibial length, there was a significant increase in the heart rate. ANP, BNP are the marker for the heart dysfunction. Their secretion as well as expression in the myocyte was significantly increased and was attenuated by WFA. We could not see much difference in MHC alpha, but MHC beta, which is a fetal form, was significantly increased and then reduced in WFA treated animal. Fibrosis of the heart is another, and we did H&E staining and then Mason trichome staining, and we could see at the perivascular, there was a deposition of fibrosis significantly. You can see from the analysis here, and WFA attenuated the increase in fibrosis, reversed the fibrosis. This is again cross-sectional area, not using H&E staining, but different staining, I forgot the name. So we can see in a vehicle saline implant animal, there was whatever size is there, and WFA did not change its size. Whereas the angiotensin II implanted animal, we could see increase in cross-sectional area or cell size, and which was decreased or attenuated by WFA treatment. So here is the quantitation, which shows or confirm the hypertrophy of the heart. So we look for the heart function. So if you look here, LAX, LVPW, which is a long axis, it was increased because of the hypertrophy of the heart, and then decreased by WFA treatment. Left ventricle fraction shortening, long axis was also decreased, then attenuated, and left ventricle's fraction shortening on the short axis also decreased. So that clearly shows the damage to the heart. And these are the other parameter, velocity of the pump, it was decreased. And this is again left ventricle inner diameter, and EA is diastolic function, which was also changed. E to A ratio was changed. And then LAX, this is diameter again, was also changed. So if you look here, I will show you what we saw in animal that those were implanted with the angiotensin II pump. So this is a control animal. An animal mouse has about 400 to 450 beats per minute. So this was beating normally. And WFA did not make any difference. So it was beating normally. And now we see here, this is the heart, which animal which were implanted. See, it stops, its contraction are stopped, and heart start vibrating. And we saw in almost every animal. So I want to show again, look here, see, now it's vibrating. The contraction is gone down and the heart is vibrating, which is showing the damage to the heart or dysfunction of the heart. And when we treat it with WFA, the normal function was retained or gained. So that's what we shot here. Okay, next. Okay, so these are the inflammatory cytokine which were upregulated. IL-18, we didn't see much difference or no change. IL-17 was upregulated, interferon gamma was upregulated, and MIP2, which is IL-18, was also upregulated in angiotensin II implanted animal. So we looked the inflammasome again, which is Dr. Radachek's favorite molecule and became my favorite interest. So NLRP3, caspase-1, ASC, and MHC beta. So all were activated, and this confirmed the immunostaining of this molecule, NLRP3, ASC, and caspase-1, and this is the quantitation by using the scanning. So everybody in the field works on left ventricle. They see the damage in left ventricle and they study left ventricle. Why? Because left ventricle pumps blood to the whole body, and they think that this is the most important function. Right ventricle, nobody pays attention, except for the last few years, people have started paying attention. The left ventricle pumps blood only to the lung, so they don't think it's important. So you don't see any drug for the left ventricle damage, but you see number of drugs for the left ventricle damage, a lot of drugs. With the right ventricle, there's no treatment. I talked to some cardiologists. They say that if patient comes with a right ventricle damage or dysfunction, we say, okay, live with it. We don't have any drug. That's true?
Most of our RV dysfunction is actually pulmonary hypertension.
Yeah.
So there's a whole array of medications, infusions to try to deal with that high mortality rate. But your point's well taken. Doesn't matter what the disease is, if the RV is dysfunctional, the mortality
It goes up.
tremendously.
Yeah. So what we saw when we looked the right ventricle while we were doing the left ventricle, we saw great damage to the right ventricle. There was a hypertrophy, the diameter was increased, and fraction shortening and everything was messed up. So this replicated what we saw in left ventricle. And we know the left ventricle, as Dr. William has said, it causes hypertension and also fibrosis in the lung, which cause breathing problem and other dysfunction of the heart. So we are working on this project. We have not finished yet. So we did H&E staining and then Mason trichome staining for the fibrosis. If you look vehicle or saline infused animal, we didn't see any sign of fibrosis or very small, which could be just a nature of the line. And when we infused with angiotensin II, we saw tremendous amount of fibrosis. I don't know how these animals live. This is one of the slides I'm showing, but in some tissue, we saw the whole area was completely blue. So this shows great damage to the lung. And then when we treated with WFA, very interesting, it was reverted. We reverted the fibrosis. So what's the mechanism? We are looking like fibronectin and some of the marker TGF beta, which induce fibrosis in the lung, and CWFA is reverting those mechanisms. So if we put this thing together, so we have angiotensin II pump in the animal, which increases angiotensin II circulation, and it affects the heart and induce inflammatory cytokine and activate the inflammasome, activation of IL-1 beta, IL-18, which affects the heart function, left ventricle as well as the right ventricle. The net result is heart rate is increased, wall thickness is increased, contractility is decreased, hypertrophy is increased, fibrosis is increased, and fraction shortening is decreased. These are the net results what we analyzed and found out. So conclusion for this study is angiotensin II infusion results in development of muscle trophy and cardiac hypertrophy. So this is a reverse effect and cardiac dysfunction that may make cachexia. WFA treatment attenuate loss of muscle mass and reverse cardiac dysfunction. WFA reduces the level of pro-inflammatory cytokine induced by angiotensin II. WFA reduces the level of inflammasome cardiac muscle induced. Most important thing, what angiotensin II treated with WFA does not have tumor. So the effect of WFA is a direct effect. So I believe not only the cancer patient who develop cardiac dysfunction can be treated, but the patient who get hypertension and other cardiac dysfunction can also be targeted by WFA. That's my theory, but we need to check. Okay, this is a dream of every professor. What your theory formulation or what you have done in the lab, is it applicable to human? So in my study, I studied the combination of doxorubicin, doxil, cisplatin with vidofludimus. What we found out that when we use the combination, the effect is synergistic. It reduces the doses of each agent and and reduces the side effect. So with that, I wrote a protocol, IND application, and submitted to FDA. And fortunately, both phase 1 and phase 2 are approved by FDA. So we are at the stage of recruiting patient, and this patient will be treated with doxil in combination of WFA in the form of ashwagandha. Doxil, we use the standard of care, 40 milligram per meter square, and we have phase 1 and phase 2. Phase 1 will have 18 patients, and phase 2 will have 54 patients. In phase 1, we will use the 3 doses of oral administration of vidofludimus in the form of ashwagandha, and we'll find out which dose works better. We start with 2 milligram, then go to 4 milligram and 8 milligram daily, twice daily. And then standard of care is a 40 milligram per meter square, which will be administered every 3 to 4 weeks. I think it's 4 weeks. And what we are looking, as we said, that doxorubicin itself causes myocardial toxicity. So it may be possible that it may, with different combination, may revert myocardial toxicity, and it may have a direct effect on cardiac function. What we are looking is the outcome, the toxicity profile, overall survival, partial response, progression-free survival, stable disease, and cardiac function. Quality of life should be improved. We have obtained the FDA approval and currently recruiting the patient. I'm working with Dr. Gorsberry in the OBGYN department. She's PI on the clinical trial with me, and Dr. Miller and Redman will serve as co-investigator. This trial is active. It's on NCT site, and we are just talking to patient and get the eligible patient enrolled in the trial. So hopefully within next few months, we may have some data. So I have a lot of people to acknowledge. Alex Straubel, who was postdoc with me, initiated this Cachexia project. Natia Clem was also postdoc. She studied the cardiac function. Partha Roy is at IIT Roorkee in India. I collaborate with him. Sumara is a clinician at All India Institute of Medical Sciences, which is one of the best institutes in India. Zipeng Hong, who is here, he helped me to do this cardiac function. And K. Lu is another associate. Whitney Goldsberry will carry on the clinical trial. Don Miller, of course, he and me know more than anybody else. We know each other almost 37 years. So when I went to UofL, he was one of my interviewers. So I followed him to come here. And his interaction has been great. He's a great friend and great mentor. Dr. Maurice Radachek, we have collaborated significantly and published a lot of papers. We share the facility, we share the building, we share the crime. Sometimes things don't work, so we are having long face and talk to each other. At the same time, we build up new projects and things. The funding for the project came from NIH. This was a collaborative project with my friend Dr. Batra from Nebraska. And then we were funded T32 on stem cell and regenerative medicine, myself and Dr. Radachek. And I was funded R25, which is an excellent grant to train the underrepresented student from JCTC and build up their career. We talked to Ovarian Cancer Awareness of Kentucky, and they have promised to fund the trial, phase 1 trial. They already have given $40,000, $15,000 first round, and last Saturday they gave us $25,000. Total is $40,000, and as we go, they will pay us more money. Brown Cancer Center reduced the cost for the trial. Of course, I'm applying grant. And these are the students. Vasa Vimuri, he did some of the work, but I presented. Nicholas, Kathy, Kusum, they're all undergraduate student. Nicholas was a master's student, no, he's in medical school. Then several people from clinical trial office, without their help, I could not do anything connecting to FDA and no compliances, and these include Alexis Hand. She's a very nice and sweet person, always responsive. Manahil Khan, Rebecca Redman, Todd, then rest of the people. We are building a new collaboration. So one of the things I told that WFA is purified from a plant. So there's a variation from batch to batch with respect to purification. And also scalability for clinical purpose. So I got in touch with Joe Burleson, who is a medicinal chemistry, and Robert Monson, who managed the competition modeling facility. And Margaret is my collaborator. So we are going to test some of the commercially available compound which have similar structure to WFA, and then go for the visual screening to find out the new compound which may be different than WFA, so that we can scale up, because WFA is very hard to synthesize. It has 28 steps and it's very unstable compound. And hopefully by using visual screening and modification of the derivative, we may come up with a drug which we can be marketable. So that's something we're looking forward. With that, I stop here and be very happy to answer any question.
So I kind of have a full page of questions, but I'll try to just limit it.
I can come back.
That's probably what we should do at some point. But I'm trying to avoid the temptation to say, Ash, what kind of forever? Because it looks like you can do so many things with this.
Yeah, it's used for diabetes, stress, and many other stuff.
So, but let me start with the idea, you have a model for AT2 damage, but you didn't do any mirroring work on the vacuolization of myocytes that happens with doxorubicin to see if that's reversible, to see if that stops. Were you thinking of doing that before doing the clinical?
Yes, so since we in clinical trial, we are using doxorubicin with WFA, and I have not done, I did very small study where I injected doxorubicin.
Yeah.
That time we were not really very much aware of the people who could do heart function.
Okay.
So that thing didn't go very well, but I'm planning to have some fund where I can treat the animal with doxorubicin or doxil,
Right.
along with WFA, and see if it reverses the myocardial.
So it's been very helpful, just because from a cellular point of view, it's so pathognomonic to see those vacuoles show up.
Yeah,
And if you could stop that, there's no limit. You'd be able to buy the university. I'm sure.
yeah, I have some friends, Shahid and other who work in cardiovascular institute. I can take their help and collaborate with them, because I studied this cardiovascular 4 years.
Yeah.
During my early postdoctoral training. Since then, things have changed, cloning have changed the whole thing. I never got back. So now I'm getting and merging oncology with cardiology.
Okay, well, that would be great, because doxotoxo is one of our biggest limitations to a great drug for cancer that you just can't take. Okay, second question has to do with do you think that the final common pathway of your AT2 pulmonary fibrosis is such that with the Ferrin A could actually take care of other types of pulmonary fibrosis? We have a big IPF problem in this area. And we have a big center and a 9-month backlog. And if you could come up with a drug that could stop this fibrosis cold, then you really will. You'll buy this place in University of Kentucky.
So I can say two tissue definitely we have looked, left ventricle and the lung. So WFA is reversing the fibrosis. We looked at kidney, but somehow we didn't hit the right area. So we did not see fibrosis in kidney. Even though we submitted manuscript last week for publication, But I believe we hit the right area, we can look fibrosis in kidney, in the cancer animal, and see if that also reverse. I looked the liver, if there was any damage by cancer, liver is big, so not every area may be affected. So we didn't see much information.
So I was going with, pulmonary fibrosis as a phenomenon. It's this,
Yeah.
autosomal dominant with incomplete penetrance. It's terrible. It's still, we don't know what's happening a lot of times. And sometimes it is just the air quality of Louisville. But the fact is, it's a major problem. If there's something about that drug that could stop that process in the lungs from showing up on the trichromosome, that would be really tremendous.
Yeah, well, we can plan to do it.
Okay, so we got pulmonologists who are
Pulmonary,
dedicated to that, so can
Yeah.
I cross fertilize you guys?
Yeah, I will talk to you, who to get in touch.
That'd be
and,
good, Mohammed Saad
expand the
group
study.
is so tremendous, so that would be tremendous. The other one's a real simple one, the arrhythmia that you showed, it actually was a bradyarrhythmia, it was a dropped beat. So I couldn't see fast enough whether or not it was just a P wave that stopped, so it was sinus node dysfunction, or it was a P wave that didn't conduct, so it was AV node dysfunction. But how consistent is that happening, or is it just, was that a one-off, or are you?
We have 10 animals,
Uh-huh.
and we saw in each animal.
Really?
Yeah.
So just.
So it's not in 1 in 10 or 2 in 10. In each animal, we saw that arrhythmia.
So it'd be good to know what part of the conduction system is
Yeah, we
working on.
probably have to repeat the experiment and
Uh-huh.
pinpoint the right area.
Or send me the tracings
Tracing out.
of the ECG and when it stopped,
Okay.
if you have it. All right, so anyway, so many more.
So you keep saying reversal of fibrosis and reversal of these things. Do you know if it's reversal? Like, did you show data that's, because you only showed like one. The only one that I saw that looked like might be reversal was the grip strength at the very beginning. Because they had the week and drink and then, but everything else was constant treatment and then basically endpoint. So.
This is an excellent question,
Mhm.
and we did not have enough animal in the groups, so we could kill every week or every 3 days. So we do not know is it prevention or treatment,
Yeah.
right point, at this point, we don't know.
Yeah, I mean,
So we
I'd be
probably have
surprised if
to
it was
Have
treatment of
different groups of animal, not treat after 1 week,
Yeah,
let them
exactly.
get cancer. And then treat with WFA, by the time they have built up
Right.
all the fibrosis
Yeah.
and all the.
No, I mean, that was the experiment I was thinking of when you were talking about the reviewer's question. That seems like the most straight, test them and then put in, once they have it, then test, so.
No,
Not coming up with the whole.
that was one
angiotensin
of the
thing.
specific game in the grant,
Yeah.
that will differentiate between prevention and treatment.
Mhm.
But grant did not come through, so we did not do.
Yeah.
Yes, sir.
So we know there are a lot of commonalities and pathways between malignancy and cardiomyopathies, whatever they are, right?
Yeah,
And so one of those that gets focused on a lot is this NLRP3 IL-1B pathway, right?
Yeah.
Which seems that WFA really targets through that pathway.
yeah,
We also know that in the CANTOS trial in cardiology, and canakinumab blocked IL-1 and improved outcomes in heart failure. It looked like it might have actually caused a little bit of remodeling and things, beneficial remodeling, things like that. So my question then would be, are you just reinventing the wheel for blocking IL-1? Should we just use an already available drug to do this?
I read some paper on IL-1 beta as well as IL-6, which are the major cytokine, inflammatory cytokine. The people have used the antibodies, but they didn't see much effect.
So
Didn't
I
see
think it'd
much
be a great
reversal.
experiment for you, like a side project, take some of these same animals, the
Mhm.
same setup,
Yeah,
and use canakinumab,
Yeah,
right?
yeah.
And see if you get the same result. If you do, then the question is, why are we reinventing the wheel?
yeah, that's what the clinical trial will tell, I think, more than what we can expect from the animal model. So if we can reverse the heart dysfunction in cancer patient, these are recurrent ovarian cancer patient, they already have probably the heart problem. which
Or
is
you would
starting-
just use canakinumab in
Yeah.
cancer patients with cardiac cachexia is what I'm saying.
Yes, problem is I don't have much funding or no funding, that limits my.
But it's an opportunity. But that's the whole point, that's the opportunity.
Yeah,
Very nice presentation. I just have a comment, like, when you give WFA in the angiotensin model, you see the preservation of the cardiac function. Do you think the reviewer will come back and ask you the same question, that you see the preservation of cardiac function, so why are you looking at a muscle wasting? Because if cardiac function is preserved, obviously, there won't be any muscle wasting.
muscle wasting in
In the
skeletal
end-
muscle.
Yeah, in the angiotensin.
Well,
model.
that's just to prove the model is working. Okay, because angiotensin two also cause cardiac. Cachexia, not cardiac, skeletal muscle cachexia.
Yeah.
So to see that your angiotensin two is working, pump is working. So just after 1 week, we saw the decrease in skeletal muscle strength and the mass. That was just to test the model is working. We may not, we have separate publication on it, but not putting together. It will be just a preliminary data that shows the skeletal muscle cachexia weakness. Yeah, Mouse.
Great work, Dr. Kakar, and since you are now in, say, going into clinical trial,
Yeah.
have a lot of basic,
knowledge.
components figured out. When you develop drugs, the PK and PD and the nature of this compound itself, people have been taking ashwagandha through oral,
Mhm.
kind of supplementation. You injected intraperitoneal. The final formulation of the so-called drug, whether it is from virtual screening or molecular modeling, let us say you hit some compounds which is synthesizable, which is kind of, purity wise. So you would rather prefer it to be injected parenterally or going through the oral route?
I will prefer oral, because once you have injectables, you can reformulate as oral drug. There are companies who can do for you.
But there are questions, because when you take
Yeah, it
all
will
the
be stability
biotransformation,
and
gut
reabsorption
microbiota,
and all those things will be the question to test. That's why business grant will have to get those kind of questions. But in clinical trial, pharmacokinetic tissue distribution, of course, we cannot do tissue distribution, but we can have circulation in the blood or excretion in the urine. All those pharmacokinetics we will be looking. Yes, sir.
Yeah, I actually wanted to ask, answer to this colleague who raised some concern about interleukin-1 beta, etc. So, vidaferin, what Sham is doing, is very strong inhibitor of an NLRP3 inflammasome. And then NLRP3 inflammasome is not only interleukin-1 beta and interleukin-18, but does dermin pores in cell membrane and releases many different alarmins, which are involved in inflammation. This means, it is much broader application, not only to block interleukin-1 beta, which is one of the components, but just to inhibit overall NLRP3 inflammasome with vidaferin.
Yeah, that's something we need to plan around. Yeah, we have a gaps in the grant cover, but we didn't do anything.
Yeah, this colleague unfortunately left, and I didn't time to.
So just one.
Yeah, it
small
was a great
question.
learning.
I mean,
Yeah.
like, based on my experience with the you are planning to do clinical trial with ashwagandha.
It's
Correct.
this ashwagandha is not a supplement which you find over the counter.
Of course.
This is especially formulated. I work with a company to provide the maximum amount of vidaferin it could have.
Okay, so but it has other at least 6 within a while, correct?
Yes, yes. It
So
has,
how-
actually, there are 30 more.
Yeah, yes, exactly. So how you are gonna extract the, isolate the effect of this vidaferin when you are doing, all your study that.
That's a relevant question, and I appreciate you ask. We cannot eliminate those one in the formulation we have, but if you look the most biologically active compound is vidaferin in ashwagandha. The other content, if you look the chart, I got analysis for other vidanoid also. They are much less than vidaferin. So they may have minimum effect or no effect. That's what I assume. But minimum effect could be maximum.
Yeah, some within a while say and.
Yeah.
Indeed, they are.
But
very
once we have the
active.
synthetic compound, that will
Yeah,
be purified and. Any other question? Yes,
Are there any other plants that produce this compound?
this vidaferin.
Yes.
I have not come across. Ashwagandha is the only plant which has a maximum amount and biologically active. I did not screen all the natural plants, so really I can't answer you.
Yeah.
Maybe we can run the PubMed and see.
Yeah, the ashwagandha, the regional research laboratory in Jammu, India, they have come up with different plants. They have some what is some genetically modified plant which has very high content of this betaferrin. So that's what like few labs are trying to extract from those, and they have patented it. So
Yeah, even the Patanjali, they extract from different plants. I don't know what plant, but they don't disclose that. I started collaboration with Patanjali, but didn't go through because I didn't like those scientists.
Just last question, and
Sure.
you touched a nerve saying that you don't have the biodistribution. That would be the kind of a nuclear medicine kind of thing. So we are getting another PET center here, or PET camera,
Mhm,
and all you need is carbon-11.
mhm.
I'm looking up the chemical structure of all the withanolides have so many unsaturated ketone rings. You
Yeah.
should be able to label that thing and tell you exactly where it goes,
Okay.
and exactly what quantity
Sure.
in each species. So
Actually, people in the field using ashwagandha, they have seen the distribution in different
Okay.
tissue of WFA.
I see.
They
So
have,
you do have some.
but
idea.
those trials are not well planned and well done,
Mhm.
so I don't trust those data.
So-
But we have to do ourselves in patient. Of course, we can do the tissue, but in animal, definitely we can do.
Right, well, I mean, and that's the advantage of PET,
Yeah,
carbon-11,
yeah.
if we have a labeled. And radio labeling is not difficult, we just have to have a
Yeah.
chemistry lab and say you do it. You do a test dose that's radio labeled, do the biodistribution, and that'll help you design the trials, because, you know, I mean, if you.
We have to modify the trial, to give it
Exactly,
sea level.
something to think about.
Yeah.
As is every week, something to think about. Okay, thank you, everyone, and Okay.